Welcome back. How was spring break? Good? Y'all feel rested? Meh? Aw. Need another week? Fortunately, you don't get another week. That's not on me. All right. Who left the country? 2 people? Where'd you go? Oh. Okay, well, at least you're honest when you're called out on it. Okay. Somebody leave the country in the back? Yeah, where'd you go? Cancun. Nice. Anybody leave the state? Where'd you go? California. Where? Back home. Okay. California, back home. All right, I went to California. Who went to California? Arguably, it's out of the country. I'm just kidding. What'd you guys do? Anything fun? Let's hear it. You guys are shy. Wow. I mean, we've been together for like half a semester. Are you shy? Skiing. Anybody ski in town? Anybody get that Friday, Saturday storm? I missed it, and I got stuck not being able to get back in town. But I was surfing, so can't complain. But I wanted to get snowboarding in, right? Yeah, that was the goal. I boarded before we left, surfed, and then I wanted to get that fresh powder, but I missed it. They shut down the interstates. Anybody else do anything fun? Went to a rave. Okay. I did not do that, but I bet it was fun. Right on. Probably wasn't, hound's mouth or anything like that. All right, cool. You guys ready to get back into it? Let's start with something simple. Ready? Simple questions. What do you get when you cross an elephant with a fish? An elephant with a fish. Swimming trunks. What did the pen say to the pencil? What's your point? Oh, okay, all right. You guys are going to take some time to warm back up to this. They used to be funny, then you got a week off of me, and now they're not funny anymore. Why are frogs so happy? Because they eat whatever bugs them. Okay. What's the difference between a jeweler and a cell biologist? A jeweler and a cell biologist. One sells watches, and the other one watches cells. Come on, that's a good nerd joke, no? Oh man, that's like one of the best nerd jokes. Okay, all right. If you don't understand, stay after class, I'll explain it to you. Okay, so all seriousness now. Got to get back into it. You all see your midterm grades? Those are posted. It does not take in consideration that we're dropping anything yet on the midterm grades. It's just like a straight score. We dropped the lowest exam. And we're going to drop the lowest 2 chapter quizzes, of which you have one due this Friday. Quiz 8 is due this Friday. And a week from Wednesday is exam 3. So it's going to happen on a Wednesday, not on a Monday. Does that make sense? Okay, just making sure you guys are tracking with that. All right, so in lab, we are a little bit in front because we're going to start the central nervous system, which is the brain and the spinal cord. You're not going to see it because you're going to be in muscles and lab for a little while. So you're not going to see it for a couple of weeks. So we're getting a little off sequence, but we're coming in front, and then lab will catch up. Does that make sense? What I want to show you straight away is a little movie trailer that kind of, I think, nicely characterizes, describes our fascination with the brain. As we're going to be talking about the brain for the next 2 lectures, and then we'll get into the spinal cord. So the brain and the spinal cord together are the central nervous system. And then after exam 3, we'll get into the peripheral nervous system. And the peripheral nervous system is what's going to interact with your muscles. But the central nervous system kind of controls the muscles or receives feedback from the periphery, from sensory information about what's going on in your environment. So this is like our central processing unit. And we as a species have been fascinated with this for a while. And they made this really kind of weird, kind of cool movie that I want to play for you.
See that guy? That's me. My excuse for looking like this? I'm a writer.
Eddie, maybe it's time to let the writing go.
But just in case you think nothing ever happened to me. Eddie Mora! Hey, tell me about this book. You know how much you do right now? Not one word. Well, I suppose I can help you with that. You know how they say that we can only access 5% of our brain? This lets you access all of it. If a clinical trial is FDA approved.
That guy seems trustworthy, doesn't he?
That's all. I was blind, but now I see. A tablet a day, and I was limitless. I now had cultural appetites.
Since when do you speak Italian?
I finished my book in 4 days. I'd like to renegotiate my advance. Math became useful. From 12,000 to 2.3 million in 10 days. I'm baffled by this guy. So, Eddie Mora, you do know you're a freak. What's your secret? Medication. Your powers are a gift from God, or a whole lot of hell over your life script. I'll open up a line of credit for you if you're wanting a few toys. Not many of us ever know what it is to become the perfect version of ourselves. What's the asking price?
12.5.
I'll take it.
All right, you get the idea. So it's kind of an interesting movie. I found it very entertaining, and you can probably stream it. Couple false things, right? That's the nice thing that Hollywood does. They take a couple of interesting concepts and then they elaborate like crazy. We actually use more than 20% of our brain. It's not that 80% of our brain isn't used, but arguably not all of the brain is used. And there's areas that have neuronal machinery that we really don't know what it does. Most of the brain is actually made up of interneurons that connect certain parts of the brain to other parts of the brain. So we're going to be studying regions of the brain. We're going to be studying what, the front part does, the sides, the back, the brain stem, and as you work your way down into the spinal cord. And it's packaged and organized for all sorts of different activities, whether it's vision or sensory, or whether it's muscle coordination or movement, whether it's memory, short-term, long-term, emotions, pleasure, homeostasis, higher intellectual thinking, planning, concept of time, right? Where are you going to be in 10 years? Where were you last week? I mean, most neurophysiologists do ultimately believe that we're the only species that can operate in this concept of time. We don't have a counterpart in the animal kingdom that really can conceptualize how was spring break and what you do last week versus where do you think you're going to be in 10 years? What's your 10-year plan? What's your 5-year plan? Some of you are like, heck, I don't even know what my plan is next week, okay? But those kind of concepts are what make us human. And so this section is actually really kind of cool. And we're really, this is a general 201 anatomy physiology course. So we're just getting into the surface. You move on in your coursework, in your studies, grad school, med school, PT, etc., PA school, you'll be taking neurophysiology courses. And this will be kind of the foundation of neurophys. I have a good friend who's a PA. He's a neurosurgical PA. So he does a lot of surgery with neurosurgeons, but he does the surgery as well. And he's a physician assistant. And a lot of what he deals with is head trauma and accidents like skiing issues, car accidents. All right, I understand one of the reasons they shut down all the highways and freeways in and out of Flagstaff Friday and Saturday was because of a lot of the road conditions, right? There was a horrible accident on I-40 and a few fatalities, which is devastating. And so a lot of that comes to the trauma center that's nearest by, and that's Flagstaff Medical Center. Okay, so all of this kind of stuff is what we get to unpack over the next 3 lectures. Central nervous system, brain and spinal cord. So let's focus on the brain. We're going to talk about the brain for the next 2 lectures, and then we'll get into the spinal cord. Some general features. Well, there's 2 hemispheres. There's a right and a left. We'll talk about hemispheric specialization as well, because certain parts of the left hemisphere operate certain things. Likewise, certain things in the right hemisphere operate other things. We both have 2 hemispheres, or all of us have 2 hemispheres. They're connected by a main white matter track of axons, or like electrical wiring, and that's called the corpus callosum. That's that main connection between the left brain and the right brain. Think of them like 2 different computers, like a dual core processor. So you have the left side and the right side, and they actually communicate. And they communicate using this corpus callosum track. We also see, and you're going to get into this in lab in a few weeks, you're going to do a brain dissection. You'll do a cow brain dissection. No, sheep brain dissection, sheep brain dissection. And you'll see that there's these bumps on the brain. And you know that. You've seen pictures. Well, these raised portions are called gyri, or singular is gyrus, and the valleys or the invaginations are called sulci, or a sulcus for singular. It's a fold. What is the purpose of that anatomical characteristic? What do you think that? Why isn't it smooth? What does this do for us? I saw that. Can you say that out loud? Maximizing surface area. Right? So the more wrinkly your brain, the more brain matter you have, the more intelligent the organism. So by having these folds, if you laid it all out and stretched it all out, it wouldn't fit inside of our cranial cap. So we have these folds to pack more neuronal tissue into our skull. Gray matter versus white matter. So the big difference here is white matter has myelinated axons. Myelin is produced by a cell. It's a glial cell. And its job is to actually take its phospholipid bilayer that has mostly fat with embedded proteins and wrap the neuron. Like if you had wiring out of the wall and you pulled the wiring out, it wouldn't be bare wire. It would have a casing on it, right? So the myelin is like a casing. And the myelin sheath, what it does, and we'll get into this later, it actually speeds up the action potential or the transduction of electricity. So the signal goes faster down a myelinated track than it does a non-myelinated track. So gray matter is unmyelinated. So it looks gray versus white matter has this shimmery, glistening surface because of this phospholipid bilayer that has the proteins embedded in it. Why the difference? Why not just make everything white matter? You just told me that white matter is faster. Don't I just want fast? Everything fast. Why won't I just make all of the neuronal connections in the body white matter? Why is it designed that way? Any ideas? I know, it's the Monday after spring break. Does it have anything to do with how we process information or moving a body part? It does. So if you're moving, let's say, your big toe, and you want to control, everybody think about wiggling your left big toe right now. Ready? Think about, don't do it. Oh, come on, not yet. Now wiggle. You guys all wiggling the left toe? Now wiggle the right toe. How far away is that from here? That's almost the longest distance it can go, right? So which track system would you use to send that toe wiggling signal quick? white matter. If you have interneurons from the right brain moving to a right brain center, and it's maybe an inch away versus whatever, 5.5 feet, do I need to spend the biologic energy making white matter neurons right here, from here to here? I can get away with using gray matter. So you conserve biological energy. That's why we have gray matter. Is you can't make everything white matter. Everything can be platinum plated, right? So you save it for the stuff that you really need it for. Okay, so look at this picture from a lateral view. You'll see the anterior side is to your left, the posterior side is to your right. The superior portion is obviously up, and the inferior portion is where the spinal cord comes down at the bottom of the screen. So this is an orientation that we're going to be looking at all this semester, or for the next 3 lectures. Now, a gross anatomy view, gross meaning macroscopic. You can see it with your own eyes. You don't need a microscope. This longitudinal fissure separates the left from the right hemispheres. And what connects the left and the right is that corpus callosum. You can't appreciate it unless you actually have a cross-section through the brain. You can't see it. The extensive folds increases the surface area, so nicely done. And you kind of have a magnified view here of the gyrus and the sulcus, where the gyri are the ridges and the sulci are the depressions. So we pack this thing in the cranial cap, and we need to protect it. Right? So we come with basically a built-in head protection or helmet. Well, why do I have to wear a helmet when I do all these different activities, right? When I'm skiing, why do we advocate for that? Well, this isn't bulletproof by any stretch of the imagination, and there's a lot of trauma that can happen. And so we'll get into some of those things as the lectures ensue, right? First, usually we talk about how it's normally arranged and why it is the way it is. And then we talk about some of the things that go wrong. Well, we have this protection, these 3 layers known as the meninges. These meninge layers cover the brain. They cover the brain and they cover the spinal cord. So the brain, as it extends inferiorly to the spinal cord, is contiguous. And you see some of the same architecture as you work your way down. And what covers it from the outside in is dura, arachnoid, and pia. Dura mater, arachnoid mater, and pia mater. 3 different meningeal layers. And you can kind of appreciate here, here's the skull cap. This first layer is the dura. And then you have this next layer, which is the arachnoid. And then you have the 3rd layer, which sits right on top of the brain, which is the pia. The area right below the arachnoid mater and above the pia is the subarachnoid space. And that subarachnoid space that you can appreciate here on the right side of the slide is where your cerebral spinal fluid lives. So the cerebral spinal fluid, we're going to get it to in a minute, but it's basically filtered blood plasma. So it's clear, colorless, odorless, has a lot of electrolytes, no red blood cells. It's clear, it looks like water, or kind of like a murky water, if you will. And the whole point of it is to kind of bathe the central nervous system. So essentially, the brain and the spinal cord float in fluid. Kind of like apples floating in a basin of water. You know, like have you ever bobbed for apples? You can see the apple kind of floating on top. Your brain and your spinal cord are sitting in fluid. So we'll come back to that a little bit later in the lecture. So this meningeal layer, and specifically the cerebral spinal fluid, this fluid actually circulates throughout the central nervous system. And we've got a network of chambers. We call them ventricles, 4 ventricles. We have a right and a left, lateral ventricle. Then we have a 3rd and a 4th ventricle as we work our way down. And in each of the ventricles, so ventricle is a word that means like room or chamber. So in each of these ventricles, the choroid plexus, which is a vascular architecture, is what filters blood making cerebral spinal fluid. So that's where it comes from. So architecturally, Let's start with this picture on the right because this is the same orientation that we started with. We've got the top superior, we've got the bottom inferior, we have the posterior aspect to your right, and we have the anterior aspect to your left. And this blue shaded structure that's inside, now we do a rotation to the anterior view. Here is the left and the right lateral ventricles. This is the third ventricle. These two connect and basically empty into this middle third ventricle chamber. And this empties into the fourth ventricle and then down the central canal, which is the spinal cord. And the fluid that's made in each one of these at the level of the choroid plexus is made here in the left lateral, the right lateral, and then it's made a little bit more in the third ventricle, added to it, made a little bit more in the fourth ventricle, and then it comes down, and as it comes down the central canal, it's coming back up into that subarachnoid space, and then ultimately it drains into the arachnoid villus, which is dumping into the venous circulation. So it returns back to the bloodstream. So it comes from the blood, and then it goes back into the bloodstream. Does that make sense? And it's a very small amount, about 150 milliliters. That's it. 150 to 250 milliliters. That's about how much you have. Okay, so very small amount, very important. And the blood filtering that happens in each of these ventricles, there is a barrier between the bloodstream and the brain. It's called the blood-brain barrier, the BBB. This blood-brain barrier is usually impermeable to many things. Certain drugs will actually cross the blood-brain barrier, and they'll end up in the cerebrospinal fluid. Certain types of pharmaceutical agents. Certain types of viruses or bacteria or fungus can make their way past this blood-brain barrier and into the cerebrospinal fluid. And then what we do to test if there's an infection, we'll get to this a little bit later, is we do a spinal tap into that subarachnoid space, and we pull out a little bit of fluid and we test it to see what's in it. Okay, so we'll get back to that in a minute. The ventricle system. Okay, so there's about 150 to 250 milliliters at any one given time in this of cerebrospinal fluid in the ventricular system in the subarachnoid space. It's clear, colorless fluid that kind of bathes the central nervous system, creates some buoyancy so the brain kind of floats, provides some protection, it cushions the brain, and actually chemical stability because that fluid actually provides nutrition and actually neutralizes metabolic waste products. Now, if you have a situation of dehydration, severe dehydration, right? So alcohol is a diuretic. You understand what that means? It makes you pee. So you lose a lot of water volume and you don't replenish it with pure water. And so the next morning you have this monster headache because you have low amounts of cerebrospinal fluid from the night before and your brain is kind of resting in your cranial cap in spots that it's not supposed to be touching. And so you're getting pressure on the brain leading to the hangover. Right? So all sorts of different strategies of drinking water or orange juice or getting an IV or having a liquid IV, right? A lot of those things actually will work because you're just rehydrating the body pretty quickly. So how does it circulate? So similar picture that we've been looking at. We're going to start here at number 1, and we're going to end up at number 8. So number 1, the cerebrospinal fluid is secreted by this choroid plexus in each of the ventricles. We're going to start with the lateral ventricles. It flows from the lateral ventricles through this intraventricular foramina to the third ventricle. And in the third ventricle, the choroid plexus adds more fluid, like we mentioned before. And then number 4, it flows through the cerebral aqueduct into the fourth ventricle. The fourth ventricle adds more cerebrospinal fluid, like we said before. Then it moves out of these lateral apertures and fills the subarachnoid space as it makes its way back up to the arachnoid villus, it dumps into the venous circulation. Same thing I said on the slide before, just 8 different steps. Now let's do it even simpler in 6. Same information. Choroid plexus of the lateral ventricles produces cerebrospinal fluid. It goes through a hole, the foramina, the intraventricular foramina, between the ventricles. The hole between the ventricles is what that means, and into the third ventricle. And then it goes through the cerebral aqueduct into the fourth ventricle. And of course, there's all these different names for it, but the most well accepted is the cerebral aqueduct. In the fourth ventricle, more fluid is added here, and then it dumps into the subarachnoid space between the arachnoid and the pia, back into the circulation. So why are we spending so much time on this? Well, there are some disorders like hydrocephaly. This is a ventricular disease. Hydro meaning water, cephalid meaning head. And this is a swelling of the head that's caused by an overproduction or a blockage of drainage of cerebrospinal fluid. Now, it's far less common in this country because we have a lot of prenatal and postnatal care in the US, but in developing countries, the prevalence is much higher than in the US. But it's very, very serious. Like if it's left untreated, over half of these infants will pass. So what's going on is you're either producing too much fluid and or you're not draining the fluid back into the venous circulation. So you have a buildup of pressure on the brain, and because the cranial cap will prevent swelling going this way. Like you sprain your ankle and you look down and you're like, holy cow, my ankle is like 3 times the size it normally is, right? It can swell because there's no bony structure on the outside keeping it in place. But in the cranial cap, if the brain swells because of an increase in cerebrospinal fluid pressure, swelling is restricted by that cranial cap, and so you put pressure on structures of the brain. Well, obviously, in young infants where many of those fontanelles between the flat bones and the skull that we learned about haven't fused yet, the head can actually really swell. In you and I, it would be horrifically painful because all of our fontanelles have closed, and so there wouldn't be any forgiveness. So about a third will recover with normal intellectual function. So this is why we screen for this in pediatric medicine quite carefully. So I'm going to show you a case study that's a little difficult to watch, but I want you to understand how real these types of cases are, these situations are. So this is in Tripura, India. This is Runa Begum. She was 18 months old, and she was suffering from hydrocephalus. It's far more common in infants than it is in adults, but it is definitely something that can happen in any circumstance or any age. So Runa's head grew to a circumference of about 94 centimeters, which is about 3 times the size of a normal baby's head at 18 months. Her mother and father couldn't afford much. Her father was a bricklayer and earned less than $3 a day, $3 US a day. And so for about a year and a half, they took care of her on their own. And some journalists, there were 2 Norwegian students that were in the area, took some pictures and created a crowdfunding website in order to try to raise $1,600 US for the surgery, which is basically a shunt surgery. So they go into the subarachnoid space and they put in a shunt from that subarachnoid space back into the venous circulation to encourage the drainage of fluid. It's actually a very straightforward procedure for a neurosurgeon, but it's only like $1,600 US in India for this to be done. Well, they raised about $60,000 US, and that was in about 2 months. So she had the surgery. This is how big her head got. I know. And this was the day that she went into the hospital. This is post-surgery, looking really good, right? You can see that her head has shrunk down significantly. Post-surgery about 9 months later. Now, unfortunately, the case study didn't have a great ending, because remember how I said that 50 to 60% mortality for those that are left untreated. She lived to her 5th birthday and then passed. Yeah. So many of you are going into medicine. Some of you are looking at like Doctors Without Borders. You're going to be international. Okay. This can happen in the States, but it's a little bit more rare. But this is the kind of stuff that really does take place on a regular basis. So this was back in 2015. So this is about 10 years ago when this case took place. Okay, next up, meningitis. So remember how I said certain things will pass through the blood-brain barrier and enter into the cerebral spinal fluid? Well, if it happens to be bacteria, fungus, or a virus, it can actually lead to an infection in the central nervous system known as meningitis. It's actually pretty common. It's actually very common at universities, especially in crowded living quarters like dorms, okay? And in military barracks, where there's really a lot of people living together. So there's just a lot of stuff happening, a lot of bacteria and a lot of viruses and a lot of fungus, and people are stressed. They're under a lot of stress. Maybe they're not getting enough sleep, right? And so that blood-brain barrier becomes compromised, or there's an area of weakness because it hasn't healed correctly. And so some of these things can make their way through. So the symptoms of meningitis is like the flu. Like you get a rigidity of the neck, like a stiff neck, high fever. And then what people report of their friends, like, oh, man, they were acting really strange, is odd behavior. Like they're not really themselves. They'll say strange things. They act kind of weird. And it's like, they're sort of off today, don't you think? They don't feel well. These are all the symptoms. So the cause can be, it's usually bacterial, but it can be bacterial, fungus, or viral. And if the bacteria and Neisseria meningitis or streptococcal pneumonia enter the bloodstream and then make their way into the cerebral spinal fluid, you get swelling of the brain. If it's left untreated, it's 100% fatal, but it's actually pretty easy to treat with mostly delivery of antibiotics, because most of them are bacterial infections. There are some vaccines that are available. And so, the two different types that I want you to write down. So the Neisseria meningitis, under the cause, this affects your age group, college age students. Neisseria meningitis, that's what you're going to find in the dorms. And then streptococcal pneumonia, the second one, the S pneumonia. This is the pediatric and the geriatric population that typically wrestle with this. So young children or elderly, streptococcal pneumonia. So college age in your 20s, and then young and old. Questions so far? Okay, we're going to shift gears a little bit. We're going to talk a little bit about some of the architecture of the brain. So we mentioned earlier gray matter, and we talked about sort of this biological energy that's required to actually manufacture myelinated neurons. And again, it's just like in the beginning of the semester when I talked about organic versus inorganic materials. Like one's not better than the other. Like myelinated neurons, those are better. Not necessarily. They both serve a purpose, just like organic materials, right? I mean, water is not organic and you can't have life without it, right? So it's pretty important. So I don't want you to get caught up in this concept that, gray matter is not valuable. They just serve different purposes, so they have different architectures based upon, am I wiggling my big toe? Or am I sending a signal from the olfactory cortex to my memory center? Yeah, I just said that. From your smell center in your brain to your memory center in your brain. Gray matter will send those signals because they're very close to each other. But smell or olfaction and memory are very closely linked. We'll talk more about this at the end of the semester, but let me demonstrate. So close your eyes. We're going to close your eyes. I want you to think of one of your favorite childhood memories. Okay? Has to be a positive one because we're going to go positive. It can work the other way. And now I want you to think if you can remember a smell that would remind you of that childhood memory. So maybe it's turkey in the oven. Maybe it's grandma's baked bread, or grandpa's favorite family recipe, a soup or a stew, or right? Can you all kind of think, or maybe it's a perfume. Like there's certain perfume you're like, that was my grandmother's perfume. Like that smells like my grandma, right? Or you walk into a house. You walk into your grandparents' house, and the moment you walk in, you know that you're there because it has a certain smell, right? And actually, other people may not even notice it, just you do. Your friends have never said anything. You guys know what I'm talking about? Like certain smells bring back memories. So gray matter is really important, and it actually will connect a lot of these things. So let's talk about what gray matter is. So it's a major component of the central nervous system. It's got neuronal cell bodies. It's got dendrites, unmyelinated axons, glial cells, which are supporting cells, right? They're the non-neurons. They're the other cells that outnumber the neurons 9 to 1. And it has blood vessels or capillaries. The gray matter is made up in sort of like 3 regions. We've got the cerebral cortex. That's the outer layer of the cerebrum. Then we have the basal nuclei, or the basal ganglia. They're a group of nuclei that act as sort of a cohesive functional unit. They connect many different parts of the central nervous system together. Like the cerebral cortex and the thalamus, the basal nuclei connect those 2 structures together. They're associated with all sorts of different types of voluntary functions, like learning or habitual functions, eye movements, facial expressions, emotional activities. These are all part of the gray matter. The limbic system, the last one on this list. The limbic system actually is a very complex grouping of brain centers, and we're going to talk about each one of these. And it supports a variety of functions like emotion, behavior, olfaction, smell, what I just mentioned. Long-term memory, that's the limbic system. So grandma's perfume or your grandfather's favorite family recipe, this is all the limbic system. This is the sense of smell linking to long-term memory. So really important stuff, right? Now, white matter is more glamorous, right? It's covered with a lot of biological, expensive myelin to make the electrical signal faster. We organize white matter down what we call tracks. So whenever you see in the textbook or in literature, we're looking at axonal tracks, they're talking about myelinated neurons. Bless you. 3 different types: projection tracks, commissural tracks, and association tracks. So commissural tracks, let's talk about that one first because we've already mentioned it in the first part of the lecture. What's the main connection that's a commissural track between the left and the right side? What's it? The corpus callosum. This is a white matter commissural track. Okay? It's like that USB-C that goes from this side over to this side. Okay, it's that big wire that goes really fast. Projection tracts, these extend vertically between higher and lower brain centers, like top to bottom. And they carry information between the cerebrum and the rest of the body. That was my example of like, think about wiggling your big toe. Now do it. That's a projection tract. You're going from the motor cortex all the way down to your foot. Projection tract. Now, association tract is connecting information on different regions of the same hemisphere. So, bless you, it may be like back to front. So it's not right in the limbic system, which is like this far from each other, right here, or less. It's like back to front. So that association tract on the same side of the brain is likely going to be myelinated. Okay, cranial nerves. Cranial nerves are technically not part of the central nervous system. They're part of the peripheral nervous system because they're outside of the brain and the spinal cord. So I want you to know that, but I at least need to talk about them because you're going to be doing a brain dissection. And in the brain dissection on the sheep, you're going to be identifying cranial nerves. And so I just want to make sure you understand, like, it's a little bit of a dichotomy, right? Because you're looking at the brain, the bottom underside of this is the inferior aspect of the brain. And we've got 12 cranial nerves that are part of the peripheral nervous system, but they're on the underside of the brain. But because they're outside of the brain, they're technically part of the peripheral nervous system. It's semantics, and the body, remember, I always say, the body doesn't care like, oh, well, I just left the central nervous system, now I'm part of the periphery, right? We create these arbitrary delineations so that we can build chapters and textbooks and test students on questions, right? So I at least want to talk about in here, we'll talk about them more in the periphery, and you're extensively going to be studying them in lab. In lab, you'll have to memorize them. Here, I will cover them physiologically as we get to them in the peripheral nervous system. But for sake of completeness, I wanted you to appreciate like the underside of the brain prep. When you look at it, like hopefully my class is like, that's kind of funny. I'm kind of staring at the central nervous system, but I'm identifying nerves that are in the periphery. You see what I'm talking about? So there's sensory, those that are for receiving information. There's motor, those are for performing a motor function, like a contraction. And then there's some that are mixed. They have both activities. So we will talk about, like, for example, we'll cover the vagus nerve, which is cranial nerve 10. We'll talk about what it does as we get into certain aspects of the peripheral nervous system. And I'll bring up the vagus nerve and cranial nerve 10 and how we want you to know about it, but I'm not going to have you memorize them. You'll do that in laboratory. Okay, so let's shift gears now to the different regions of the brain. And as we get to the different regions of the brain, we're going to talk about some of their different activities. And this is where I think, there's going to be a lot of memorization that's required. I think it's going to be important for you to go to SI and TA review sessions where now this picture is flipped, right? So the anterior portion is to the right, and the posterior portion is to the left. You usually can tell because of this cerebellum, right? This like wrinkly, structure is at the posterior inferior aspect of the brain. So there are worksheets that we have so that you can identify areas of the brain, because it's going to be important, because different areas of the brain, not just for lecture, but also for lab, are going to lead to different functions. And the brain is organized in sort of 3 different axes. So let me explain. We talked about one already. Right to left. Right? We have the left side of the brain and we have the right side of the brain. We're going to see here in a few slides, there are certain activities that happen out of the left side of the brain, and there's other activities that happen out of the right side of the brain. And you've heard that terminology. Oh, well, you're left brain dominant. That's why you do fill in the blank. That's why you think the way you think, because you're right brain dominant, right? Well, what does that mean? Like, don't I have a left brain too? Like, you do. All of us have both. Okay? But there are definitely things that you're good at that maybe the person sitting next to you isn't as good at, and vice versa. Maybe you're artistic, you can draw, maybe you can sing, maybe you can play an instrument. Okay? Maybe you're very rational. Math is easy. You don't need a calculator, right? So those are different parts of the brain. We'll talk about that. So left to right is the first way it's organized. The next way it's organized is back to front. Back to front. Posterior to anterior. Sensory information tends to come in the back part of the brain, and as it moves forward down through tracks, it moves to motor activity in the front part of the brain. So sensory in the back, motor in the front. Okay. The third way it's organized is top to bottom, or what we say cranial to caudal. Caudal meaning tail. Okay, so cranial meaning head, caudal meaning tail, top to bottom. So higher brain functions are at the top of the brain, like the forebrain. The forebrain. That's intelligence. That's intellectual capability. That's your personality. Your personality is a higher brain function. Like who you are, the way you behave, what makes you you, that is a higher brain function. And that's part of the forebrain. And then as you work your way down, like to the brain stem, we have autonomic centers that control heart rate and respirations in the brain stem. And so lower order activities that are like autonomic, stuff you don't have to think about, happens in the brain stem. So lower down. Does that make sense? Those are the three axes. I think if you know those three axes, it's really helpful because then you can kind of figure out where things are going wrong. And that's why we kind of study this stuff. Think about the ski accident. Skier goes out of bounds, hits a tree. The I-40 accident. Somebody gets in an accident, they're rushed to the hospital, they see you because that's where you are in 10 years. You're the clinician, you're the nurse, you're the PA, you're the PT having the recovery, and they say, well, this person had head trauma on the left side, this is what's going on. You're like, okay, well, I know what's happening now. Left side, got it, okay. Right, so these specializations and the way this stuff is set up actually does matter. It's not just this memorization. You will use this stuff as you kind of figure out where did that accident cause the trauma, the lesion, that's giving the clinical manifestation that I'm actually trying to take care of right now. Okay, so now we got to get through some of this terminology. A lot of terminology on this slide. So the forebrain. The forebrain is made up of the telencephalon, which is the cerebrum, and the diencephalon. Now, these are terminologies from the Greek. So, for example, the cerebrum, we're going to break it down into the lobes, the frontal, the parietal, the temporal, and the occipital. We will always on the exam talk about that terminology. For sake of completeness, I just want you to not be confused if you ever hear at medical school, well, we're going to be talking about the telencephalon today, and you're like, the what encephalon? What is he talking about? Right? Is that am I in a dinosaur class? So I just want you to be familiar with the diencephalon and telencephalon, meaning the diencephalon is the thalamus and the hypothalamus. Diencephalon. The telencephalon, the cerebrum, is made up of the frontal, parietal, temporal, and occipital. So in this class, we will always use these words and these words. But in the medical literature, where these come from are Greek terms, and I want you to be familiar with that. So over on the right side, you can actually appreciate that we've got the hindbrain that's made up of the metencephalon, which is the pons and the cerebellum. Here's the cerebellum, which is this wrinkly-looking structure that tells you it's at the back. And then here's the pons and the brainstem that has a lot of autonomic functions. Right? Your intellectual capability is not at the pons. The pons controls things that you don't even have to worry about. And then you can see as we move our way further down, more autonomic, less higher brain function, we get down into the myencephalon, which is the medulla oblongata, right here, and then you move down to the spinal cord. And the spinal cord is basically just like a relay center between the periphery and the central nervous system. Because as you get more inferior to the medulla oblongata, you don't really have any control activities here. You're just sending sensory or motor, or receiving sensory, or you're sending motor. So this is the organization that we need to talk about. So first up, in the central nervous system, the forebrain, cerebrum, and the diencephalon. So the cerebrum, the cerebral lobes, frontal, parietal, occipital, and temporal. Now I flip the brain again. Which direction is it? Which direction is the front or the anterior portion? On the left side of the screen or the right side? Left side. Very good. And you can see where the cerebellum is on the lower right. That tells you that that's the posterior inferior aspect. So in blue, conveniently colored in blue, and labeled in blue, is the frontal lobe. This is higher brain function. We'll talk about each of these. Then just posterior to that is the parietal lobe. Occipital lobe is towards the back aspect. And then the temporal lobes are on the side. And they do correspond to the skull flat bones that you study when you look at the cranial cap. Okay, just beneath the frontal flat bone is the frontal lobe. So the frontal lobe, higher thought, intellectual capability, intellectual function. Your personality sits here. Trauma to the frontal lobe could actually alter personality. A stroke to the frontal lobe in a loved one, post-stroke, they may behave differently. They may be more, prone to swearing, for example. They may be more grumpy. They may have different personality aspects. So the posterior border is the central sulcus. And at the very back of the frontal lobe, you can see this primary motor cortex that's shaded on this slide. This primary motor cortex, and we'll get into this more. But this primary motor cortex is basically where all of the neurons for motor output go to the rest of the body. Remember how I said one axis is sensory in the back and motor in the front? So we're in the frontal lobe, where we're at the very back aspect or the posterior aspect of the frontal lobe, because right here on the other side of the central sulcus is the parietal lobe, and this is the primary sensory cortex, which parallels the motor cortex. The motor is in the front, sensory towards the back. So all of the neurons that correspond to different parts, like your big toe exercise that we did earlier, that big toe neuron is right here. I made that up, but somewhere in here. Okay? But right here, it goes all the way down to your big toe. So there is a region that controls the formation, the motor formation of speech. So it's in the frontal lobe. You can see that it's right next to the motor cortex. It's called Broca's area. And you can see that it's getting information from a sensory area known as Wernicke's that we'll talk about next. And through myelinated neurons that go to Broca's area, and then through gray matter that goes to the motor cortex, Broca's is receiving information about speech and deciding what you're going to say and controlling the speech pattern that comes out of your mouth. Like somebody cut you off in traffic, Broca's area in the frontal cortex is the one that says, have a good day. You think about it, right? That's what you hope you do. Or Broca's is the one that says, right? Something different. So formation of speech, that's the motor component. So the next time you get in an argument with a loved one and you're like, I have so much I want to say right now, and you decide not to say it, That is Broca's area controlling the speech pattern, higher thought process in the frontal lobe, making the decision to say, I'm not going to be mean. I'm not going to say that. I'm going to choose to say something else. Or it's the one that says, let it fly. Okay? That's Broca's area. One second. So an aphasia is a term that means a language disorder. A Broca's aphasia is a disorder where if you have a stroke or head trauma to Broca's area, or if you have a neurodegenerative disease that creates dementia and those neurons in Broca's area are affected, like in Alzheimer's, then you understand what's being said because that comes from a different part of the brain. We're going to study that next, but that's Wernicke's. You understand what's happening and what's being said, but you do not allow for word production or you can't necessarily control it. So a stutter. Okay. So causes of Broca's aphasia are stroke, head injury, brain tumor, brain infections, neurodegenerative diseases, like dementia. Alzheimer's being one of the more common types of dementia in patients. Okay, there was a question back here. Okay, yeah, I was wondering if I might get to it, so thanks for holding on for me. Okay, so let's talk about the parietal lobe. The parietal lobe is in purple, right? This is the precentral gyrus here. This is the postcentral gyrus, because this is the central sulcus. So this is motor. Right here is sensory. So the parietal lobe integrates sensory information and receives all this information from all over the body. So on this postcentral gyrus, we will have a map of all the neurons and all over the body that are coming from the periphery, and ultimately the information on the inside of your sock when you wiggled your big toe is going back to the postcentral gyrus and the parietal lobe, confirming, yep, you wiggled your big toe. Nice job. You did what he said. So this information is sensory coming from the periphery, confirming your outside environment. So that axis of sensory towards the back, motor towards the front, that theme stays. That's helpful as we study this. This Wernicke's area, we also showed it on this slide, this Wernicke's area, which is intimately connected to Broca's area, but Wernicke's area, these are the names of the individuals that studied these, controls the understanding of spoken and written words. So if you have a Wernicke's aphasia, it allows for word production, but word comprehension is damaged. So you are saying words smoothly because the motor is fine, but the comprehension may be off. Or you're saying something that doesn't pertain to what was actually being asked. So you're aware that you're speaking, but you can't stop yourself and you don't really understand the words that you're saying. Same causes, stroke, headache, or head injury, not headache, head injury, brain tumors and infections, neurodegenerative diseases as well. Now, could there be something that affects both? Yeah, absolutely. Okay. Bless you. So kind of interesting, right? Like you have a patient, you have a post-stroke patient as a physical therapist, and you're working with them, and you're understanding what they're wrestling with. If it's a motor speech problem, it's Broca's. If it's a word comprehension problem, it's a little bit more posterior. It's probably Wernicke's. All right, the temporal lobe on the sides, the temples. The temporal lobe allows for auditory smell. That's what auditory is hearing. Olfaction is smell. Gustatory is taste. And this helps you interpret all of this information. This lateral sulcus is what separates the frontal and the parietal lobes from the temporal lobe. The occipital lobe. Visual acuity. So head injury to the occipital lobe, this phenomenon of seeing stars, if you flick a nerve, if you strike a nerve, you mechanically can stimulate it to send a signal. Because if you flick a nerve or tap a nerve, you can open mechanical gates that allow for sodium ions to come in. If you hit a mechanical gate and allow for sodium ions to come in, you send an electrical impulse. So when you smack the back of your head on the ground and you wake up or you get up and you see stars, right? That is the real phenomenon of mechanically stimulating a number of different neurons in the occipital lobe, and you really aren't interpreting any visual information. You're just sending signals that are artificial signals. So the cerebrum, precentral gyrus that we talked about, does what? Precentral gyrus does what? Functions to control what? Motor activity. And the postcentral gyrus, in purple on the right side of the slide, is the counterpart. Functions to process what? Sensory information. Very good. So this image, upper right, you can see the anterior portion, the posterior portion down low. This is the right side. This is the left side. This is blue frontal lobe highlighting the precentral gyrus. Purple postcentral gyrus, sensory. So motor and sensory. This is sensory on the left, motor on the right, coming from these areas. This terminology of homunculus is a Latin word for little human. So what's been mapped in the pre and the postcentral gyrus is all of the neurons that either leave or come in. If they are motor neurons, they leave and they go to different parts of the body and they control different parts of the body. So for example, the ability for motor activity, the amount of real estate that you designate in the prefrontal gyrus for the hand is large. So the ability for motor activity of the hand far exceeds that which you can do with the shoulder. Does that make sense? That's how you kind of interpret this information. It's like, I mean, you can move your shoulder, but you can't snap your shoulder, right? You can't signal the guy that just cut you off that they're number one, right? And same with the face. Look at the amount of neuronal real estate that's designated on the face. Facial expressions are huge for us as a species, right? So, and you can kind of appreciate, like, the big toe exercise. There's not many neurons that control the big toe. Like, really the only thing you can do with it is wiggle it, right? But facial expressions, tongue, hand, a lot more real estate, motor activity is designated to these types of body parts. Sensory information. Same thing here. This is a map of the different parts of the body that give you an idea of, what you can discriminate from your outside environment, what you can ascertain of information, sensory information that's coming in based upon how much sensory neuron activity you have. And the face is huge. I mean, we arguably interact with our environment mostly with our head, is what this tells me. And we'll get to this when we talk about special senses, because all of the special senses, vision, smell, taste, hearing, balance, all of these special senses, this is the last unit at the end of the semester, all of them are found here in the head. And so we interact with our environment right here. This is what we use to interact with our environment. What we see, what we hear, what we smell, what we taste, right? So that's how you interpret this information of the homunculus. It's kind of an interesting task, don't you think? Okay, the limbic system. I want to go back to the limbic system as we wrap up today in the next 10 minutes. We're going to do the limbic system, and we'll talk a little bit about the cerebral hemispheric specialization, and that's where we'll stop. So the limbic system is this structure that we mentioned earlier that controls emotion, behavior, memory, smell. And these three structures that you can see on this slide, the amygdala is right here, the hippocampus is right here, and the cingulate gyrus is right here, all part of this limbic system that gives rise to all of these physiologic activities spread throughout the brain. They're not located in one center. And mostly gray matter is connecting all this because they're very close to each other. But in contrast, the two hemispheres are relatively far apart, right? The right and the left side of the brain. So the left hemisphere has areas for language and reasoning. And the right hemisphere has areas for pattern perception, artistic awareness, imagination, music comprehension, the dreamer activities. And the corpus callosum, which is a commissural tract, white matter, is what connects the two hemispheres. And so if we look at the left and the right side of the brain, again, I'll repeat, we have both. All of us have both. But you can see that the centers for logic and facts and math and science and, reality or what makes sense, if you use those terminologies when you're talking about a situation. Stuff that's safe. Let's not take any risks on this. These are activities that are coming, ideas that are coming out of the left hemisphere. And then those ideas are crossed over to the right side. And then the right side says, well, how do I feel about that? And likes imagination, philosophy and religion, spatial perception, fantasy-based, likes to take risks, right? Your social aspects and social groups come out of the right side of the brain. So as we go through our day and our weeks as individuals, there is this constant communication of ideas from the left hemisphere and the right hemisphere coming together and making a decision about what you're going to do in a specific circumstance. And the frontal lobe, the higher brain center, is what helps to process what's my decision ultimately going to be. Right? It's sort of the judge trying to decide. And if you're honest with yourself and you look at this list, you probably gravitate towards one side versus the other in different life events, right? Maybe with school. Maybe with school, you tackle it one specific way. But like in your friend group, it's a different thing. Or at work, it's different. Or your favorite activity, like the way you snowboard, maybe it's different there. But all of you can look at this and say, like, that's interesting. Like, I see myself see aspects of me on both sides of those columns. And then you can probably, if you have a sibling, right, from the same mom and dad, same gene pool, you can look at those and go, our dominant sides couldn't be more different, right? If you have a sibling, maybe they're, really good with music and have a great voice and can carry a tune, but they're horrible at math, right? It's just fascinating how our brains are organized. So cerebral lateralization is what we refer to this as. Cerebral lateralization. And so you can kind of see some other aspects on the right hemisphere, right? You can see left-handed motor control, obviously. One thing I didn't share yet is motor and sensory both come down from the brain, and then they actually decussate or cross over to the opposite side of the body in the brainstem. And we'll talk about that. So a stroke on the left motor cortex means that the patient, right? Let's say the stroke on the left motor cortex controlled motor function of the right face. And so they have a right-sided droopy face. And their right arm can't extend. And they're walking with a limp on the right side because it was a left hemisphere motor stroke. Does that make sense? And you'll see that in patients. And you're like, why is it just on one side? Because the stroke happened only in one hemisphere. And fortunately, they survived, because not all strokes patients survive. These are surviving stroke patients. Music ability, right? Left-handed motor control on the right side. Right-handed motor control on the left side. And the tract that connects the two, this is the picture, this is the last one we'll leave with, is this corpus callosum, which is this huge commissural tract that goes from the left side to the right side. Right? I mean, if you're right-handed, you can still do things with your left hand, right? And if you're left-handed, you can still do things with your right hand. And they can communicate. You know, they can work together to open up a water bottle because they're communicating through that corpus callosum. And believe it or not, putting this cap back on that water bottle and doing that motion, there's so many association tracts coming together to make that happen. All right? All right, have a nice Monday. I'll see you all on Wednesday. Don't forget quiz on Friday.